Good day everyone. Today we will discuss on the new chapter entitled Clean Technology and Pollution Control, and how biotechnology can be applied in this field. Clean technology and pollution control are important for taking care of our environment. Clean technology means using new and smart ways to do things that don't harm the environment. It helps us save energy and use fewer resources. Pollution control is about stopping or reducing things that make the air, water, or soil dirty. These efforts are crucial for making our world cleaner and healthier, so we need to use simple and smart solutions to protect our environment for today and tomorrow. In this chapter, we will talk about different parts of biotechnology. First, we'll look at enzymes and how they help in different processes. Then, we'll discuss how biotechnology is used in industries to make things in a way that's good for the environment. After that, we'll explore how biotechnology is helping in farming by changing crops to be better and controlling pests. We'll also talk about biofuels, which are special kinds of fuels made in a more nature-friendly way. Lastly, we'll learn about biopolymers, which are like plastics but can break down easily, helping to reduce pollution. These topics show us how biotechnology is making positive changes in different areas. By the end of this chapter, students should have a clear understanding of the concept of clean technology, being able to explain how it involves using smart and eco-friendly methods to protect the environment. They should also be capable of discussing various examples of new biotechnology products, illustrating the innovative ways in which biotechnology is shaping different industries. Furthermore, students should be able to summarize the overall progress and development of clean technology, recognizing its evolution over time. Lastly, the chapter aims to equip students with the ability to assess how biotechnology is applied in pollution control, emphasizing its role in creating solutions for a cleaner and healthier environment. Let's take a look at the objectives of clean technology. Clean technology pursues a dual objective of creating more efficient and cost-effective products while simultaneously managing the negative impacts of fossil fuels on the environment. This involves the development of innovative processes and products that optimize resource usage, minimize waste, and promote economic and environmental sustainability. Additionally, clean technology strives to transition away from the environmental hazards associated with fossil fuels, seeking alternative and renewable energy sources like solar and wind power. Furthermore, the focus extends to improving the overall utilization of natural resources, ensuring a sustainable approach that reduces dependence on finite resources and fosters long-term environmental well-being. The overarching aim is to foster a greener and more sustainable future through technology that balances efficiency with environmental responsibility. Clean technology is a term that describes products, processes, or services designed to reduce waste and use less non-renewable resources as possible. It involves applying principles from biology, focus on resource efficiency, and incorporating second-generation production concepts in fundamental industries. In simpler terms, clean technology focuses on creating things and providing services in a way that reduces waste, uses fewer resources, and follows smart and eco-friendly practices, contributing to a cleaner and more sustainable environment. The figure illustrates the key sectors at the forefront of clean technology, with a focus on energy, low-carbon transport, and the production of new materials. These priority areas highlight the emphasis on sustainable practices and environmental responsibility. Additionally, other evolving sectors include the use of electric vehicles, EV, sustainable construction, and innovations in agriculture. These emerging areas signify a broader adoption of clean technology across various industries, showcasing a growing commitment to environmentally friendly solutions and advancements in technology for a more sustainable future. Now, let's discuss the first application of biotechnology in clean technology, the use of enzyme. Enzyme technology involves the application of enzymes in various industries, bringing about new and beneficial uses. These industries include food, feed, agriculture, paper, leather, and textiles. Enzymes play a role in creating cleaner products by reducing the use of hazardous chemicals. They help minimize byproducts, contributing to more sustainable and reusable processes. Enzyme technology operates under mild conditions, which is environmentally friendly and energy efficient. One of its significant advantages is the potential to reduce costs in different processes, making it a cost-effective and environmentally responsible approach in various industrial applications.
Enzymes used in industry is called industrial enzymes.
Industrial enzymes are produced from microorganisms, encompassing both eukaryotes and prokaryotes. The production typically involves a fermentation process, which can be either solid-state or submerged. In this process, microorganisms produce extracellular enzymes, releasing them into the growth media. This method makes production more straightforward compared to other approaches, as it doesn't require cell disruption.
Additionally, the extracellular enzymes simplify the purification process, contributing to a more efficient and cost-effective production of industrial enzymes. Overall, this production method utilizes the capabilities of microorganisms to produce enzymes used in various industrial applications. Several examples of industrial enzymes highlight their diverse applications in various processes. Bacillus sp, a frequently utilized bacteria in enzyme production, produces subtilicin, recognized as the first industrial enzyme. Subtilicin is particularly effective in breaking down proteinaceous substrates and finds extensive use in detergent formulations. This enzyme is produced by Bacillus licheniformis. Another important industrial enzyme is alpha amylase, produced by the same bacterium.
Alpha amylase is characterized by its high thermostability, making it well suited for breaking down starch into oligosaccharides in industrial processes. These examples underscore the significance of industrial enzymes, derived from microorganisms like Bacillus sp, in enhancing the efficiency and sustainability of various industrial applications. Other important examples of industrial enzymes demonstrate their versatility in different applications. Cellulase, produced by the fungal strain Trichoderma, plays a pivotal role in the treatment of textiles by breaking down cellulose, a major component of fabrics. Additionally, it serves as an additive in detergents, contributing to more effective cleaning. Glucoamylase, produced by the fungal strain Aspergillus, is essential for the complete breakdown of starch into monosaccharides, finding applications in industries such as food and beverage production.
Another crucial enzyme, glucose isomerase, produced by Streptomyces sp, is significant for converting glucose into fructose.
This conversion is particularly valuable in the food industry, as fructose is sweeter in taste and often used as a sweetening agent. These examples underscore the diverse roles of industrial enzymes in processes ranging from textile treatment to the modification of sugar properties, showcasing their importance across various industries.
This table shows commonly used industrial enzymes and their producers. Note that most industrial enzymes are originated from fungi and yeasts. Industrial enzymes are typically produced through a fermentation process, involving microorganisms like bacteria or fungi.
In this process, microorganisms are cultivated in a suitable growth medium, and under specific conditions, they naturally produce and secrete enzymes.
The fermentation can occur in either a solid-state or submerged environment.
Extracellular enzymes, which are enzymes released outside the microbial cells, simplify the production process as they are easily accessible.
Moreover, enzymes produced using this method often do not require the complex and energy-intensive step of cell disruption. Once the fermentation is complete, the enzymes are harvested, and a relatively straightforward purification process is employed to obtain the final industrial enzyme product.
This efficient and cost-effective production method highlights the biological advantages of microorganisms in generating valuable enzymes for various industrial applications. Industrial biotechnology, often referred to as white biotechnology, involves the application of biotechnology to process and manufacture products from renewable sources.
This field utilizes living cells and their enzymes to create sustainable solutions.
Industrial biotechnology involves in various sectors including chemicals, pharmaceuticals, food and feed production, detergents, pulp and paper manufacturing, textiles, energy, as well as materials and polymers. The color-coded distinctions, such as red for medical and green for agricultural biotechnology, underscore the diverse applications within industrial biotechnology, emphasizing its role in creating environmentally friendly and resource-efficient processes across multiple industries. Generally, the development in industrial biotechnology can be divided into three eras. Before the 1940s, industrial biotechnology was in its early stages, with the use of microorganisms for processes like fermentation existing but lacking systematic engineering.
The pre-1980s era witnessed a transformative shift with advancements in microbiology and the advent of genetic engineering.
Understanding DNA structure enabled deliberate modifications in microorganisms, leading to the commercialization of products like insulin. In the current era post-1980s, industrial biotechnology has undergone rapid evolution. Genetic engineering, synthetic biology, and advanced screening techniques have propelled the design of microorganisms for diverse applications, from pharmaceuticals to renewable energy. The focus has shifted towards sustainability, with biorefineries and renewable feedstocks playing pivotal roles, reflecting a commitment to environmentally conscious practices and innovative solutions. The industrial biotechnology value chain originating from agricultural byproducts begins with the sourcing of agricultural residues such as crop residues, food processing waste, or other biomass materials. These byproducts serve as the raw materials for the biotechnological processes. These agricultural residues is then converted through biochemical or microbial processes, using enzymes or microorganisms to break down the biomass into sugars or other bio-based chemicals. Then, the intermediate products are processed to extract desired end products. This may involve fermentation, purification, or other separation techniques. The refined products, which can include biofuels, biopolymers, or biochemicals, are then utilized in various industries like energy, materials, or chemicals. The final stage involves the commercialization and distribution of these bio-based products to end users, completing the industrial biotechnology value chain. Throughout this process, the emphasis is on creating sustainable and eco-friendly solutions, utilizing agricultural byproducts to produce valuable materials and reduce environmental impact. Industrial biotechnology is characterized by a commitment to sustainability and environmentally friendly production practices. It emphasizes reduced energy consumption, contributing to overall energy efficiency. The application of industrial biotechnology results in decreased greenhouse gas (GHG) emissions, helping fight climate change. Another key feature is the reduction in waste generation, reflecting a focus on eco-friendly processes. Industrial biotechnology is known for its highly efficient processes, optimizing resource use and minimizing environmental impact. Importantly, it often leads to cost savings, with lower operating and capital expenditures compared to traditional methods. Additionally, industrial biotechnology enables the bio-based production of value-added chemicals, offering alternatives to conventional, often less sustainable, production processes. These characteristics collectively underline the role of industrial biotechnology in creating more sustainable, cost-effective, and environmentally responsible industrial practices. A bioprocess involves utilizing living organisms, parts, or products to manufacture new products and manage waste. This interdisciplinary field combines biological knowledge with engineering skills. Key engineering aspects include the design and operation of bioreactors, sterilizers, and product recovery equipment, as well as the development of systems for process automation and control. Bioprocess engineering also extends to laying out efficient and safe biotech industries. Compared to conventional chemical methods, bioprocesses offer advantages such as operating at lower temperature, pressure, and pH. They also utilize renewable resources as raw materials, promoting sustainability, and generally produce more products with less energy consumption, making them more environmentally friendly and resource efficient. Upstream processing, UPS, involves the initial phases of a bioprocess leading to and covering the fermentation stage. This includes the development of producer microbes, where scientists work on engineering or selecting microorganisms for specific functions in the upcoming fermentation process. The formulation of the fermentation medium, providing the necessary environment for microbial growth and product formation, is a crucial aspect of upstream processing. Additionally, the design of the fermentation process, considering factors like optimal conditions, scale, and duration, is a pivotal element during this stage. Essentially, upstream processing sets the foundation for a successful and efficient fermentation process, playing a vital role in the overall bioprocessing chain. Microorganisms used in bioprocesses possess characteristics vital for safe and efficient applications. Designated as GRA, generally regarded as safe, they adhere to strict safety standards. Their ability to grow in a simple growth medium simplifies cultivation, reducing costs. These microorganisms produce non-toxic end products, ensuring safety in various applications. With rapid growth rates, they contribute to efficient bioproduction. Crucially, they are engineered to reliably produce desired products, offering consistency. Their genetic amenability facilitates manipulations for optimized traits. Resistance to microbial inhibitors enhances their robustness, and genetic and physiological stability ensures reliability over time. Easy harvest and reduced oxygen requirements make them resource efficient, highlighting their suitability for sustainable and effective bioprocesses. Characteristics of industrial-scale media in bioprocessing are strategically adjusted for maximum efficiency and reliability. These media aim to produce the highest yield of product or biomass per gram of substrate, achieving optimal resource utilization. They strive to attain the maximum concentration of the desired product, facilitating streamlined downstream processes. Designed for the highest rate of product formation, these media contribute to time-efficient production. Minimizing undesired byproducts ensures the purity and quality of the final product. Consistency is important, with these media being readily available throughout the year to sustain uninterrupted production. Their formulation emphasizes ease in preparation and sterilization, reducing complications in various production aspects like aeration, agitation, extraction, purification, and waste treatment. In essence, industrial-scale media embody characteristics that prioritize efficiency, reliability, and the consistent delivery of high-quality products. The industrial fermentation process is systematically classified based on the mechanisms governing the conversion of raw materials into valuable products. This classification considers the diverse reactions inherent in the process. Notably, industrial fermentation involves the intricate mixing of three heterogeneous phases: microorganisms, the fermentation medium, and, in the case of aerobic fermentation, air. This dynamic mixing is essential for fostering the biochemical reactions leading to the desired product formation. Unit operations, including mass transfer, essential for supplying oxygen to microorganisms, and heat transfer, to maintain optimal fermentation conditions, are critical components. The use of advanced equipment, such as multi-bladed impeller bioreactors, as shown in the diagram, enhances mixing efficiency, ensuring uniform distribution of microorganisms and nutrients for optimal productivity in industrial-scale fermentation processes. An example of bioprocess is in the production of chlortetracycline, CTC, by Streptomyces aureofaciens. Firstly, the development of inoculum occurs in seed tanks 1 and 2, where Streptomyces aureofaciens is cultivated to create a concentrated and active culture. This inoculum is then transferred to the large-scale bioreactor equipped with advanced technologies. In the main fermentation process, ammonia and glucose are introduced. Ammonia serves as a nitrogen source essential for microbial growth, while glucose provides a carbon source for metabolism and chlortetracycline production. Additionally, a defoamer is often used to manage foam formation during the fermentation process. The bioreactor is carefully controlled to maintain optimal conditions, including temperature, pH, and aeration, to promote the growth of Streptomyces aureofaciens and enhance chlortetracycline synthesis. As the fermentation progresses, the broth rich in chlortetracycline is harvested, and downstream processing involves extraction and purification steps to isolate the antibiotic. The subsequent stage after fermentation process is known as downstream processing or DSP. This phase aims to isolate and purify the fermentation products to make them suitable for their intended use. For products created inside the cells, intracellular, the processing involves handling the cells, while for those produced outside the cells, extracellular, the focus is on processing the spent medium. DSP employs techniques such as filtration, sedimentation, and flocculation to separate and purify the desired products from impurities. The efficiency of DSP is closely linked to the efficiency of USP, emphasizing the interconnectedness of these stages in the overall production process. Downstream processing typically can be distinguished into three main stages: primary recovery, product purification, and finishing processes.
In the primary recovery stage, the focus is on the initial separation of the product from the fermentation broth.
This often involves processes such as cell harvesting, where cells are separated from the liquid medium using methods like centrifugation or filtration.
Following primary recovery, the process moves into the product purification stage.
Here, the goal is to refine the isolated product further and remove impurities. Techniques such as chromatography, filtration, and various separation methods are employed to achieve higher purity. The final stage, finishing processes, involves additional steps to ensure the product meets the desired quality standards. This may include further purification steps, adjustments to the product's formulation, and processes to enhance its stability and shelf life.
This flowchart summarizes the overall bioprocess.
Beginning with USP, the initial step involves the cultivation of microorganisms or cells in a bioreactor, optimizing their growth conditions.
Following this, solid separation is implemented to obtain the fermentation broth and separate the solid biomass.
The process then transitions into DSP, where cell disruption takes place to release the intracellular product. Subsequent to cell disruption, product clarification is employed to remove remaining cellular debris and particulate matter, ensuring a cleaner solution. The clarified solution undergoes soluble product recovery, focusing on isolating and purifying the desired product from the liquid medium. Finally, finishing operations complete the process, encompassing additional steps to meet specific quality standards, including formulation adjustments and measures to enhance stability.